Hi everyone, and welcome to Unit 2. In this unit we're going to be talking about membranes and membrane permeability and membrane transport, things like channels and transporters and all kinds of things. But I wanted to start off with our devotion for the unit. For this, I'm taking one of my favorite verses because I think it's really pertinent now, and always really, but especially now. From Philippians 4:13, I can do all things through Christ who strengthens me. So I know right now we're going through some pretty interesting times, to say the least, and actually quite difficult times. For some people it's more difficult than others, and I know that courses right now are difficult for me in terms of teaching. There's just a lot of things to coordinate in terms of getting it online. They're probably very difficult for you in terms of learning through a different modality and just kind of getting everything going in this first couple of weeks. But also things are really difficult for people who might be experiencing sickness or loved ones who are sick or have unfortunately passed away, loss of jobs. There's just all kinds of stress going on. But remember, the same God who parted the waters for Noah, the same God who created you and me, the same God who made this universe and yet still wants a relationship with you and me, that same God can strengthen us through all of this and can allow us to do great things and continue through and persevere and even thrive despite the hardest situations. So I think that's really important. I also like this image because it kind of reminds me of God creating a hydrophobic barrier through the Red Sea, or through Moses, I guess, but through God's work, obviously. And there's a channel, and that's a big part of what we're about to talk about in this unit. All right. So let's talk a little bit about the membrane. This is where a ton of really, really important things happen. All right, so we can see all kinds of things going on here. We can see signaling. Extracellular signal has to bind to a membrane receptor, then you get an intracellular signal. We've got transport. We've got a channel that opens up here to allow ions to go through. We will have transporters that allow glucose through. We have cell adhesion, so membrane proteins holding cells together. All right. So a big part of it is kind of that selective permeability. And then within that, we also have compartmentalizations, we have structure, we have protection, we have localization of various aspects throughout. Energy conservation and generation. This is a little bit more difficult one, but see what you can think about that. Certainly environmental sensing, cell-to-cell communication, cell-cell attachment, and so forth. So pause the video for a second and just really take a few moments to think about what each of these means and how each of these are being performed at the membrane, or at least how the membrane has a big component of allowing these things to occur. All right, so let's talk a little bit about this. So functions, compartmentalization. This and selective permeability kind of work together, but this is probably the biggest aspect. We have individual organelles in the cell, so the membrane basically allows things to be kept in the right spot. We need the mitochondria to have various proteins and to have reactions that are occurring there, but not elsewhere. All right, and that's the same with the nucleus. The nucleus is housing the DNA, so it's compartmentalizing the DNA and preventing it from being everywhere else. The cell membrane itself is compartmentalizing things to inside versus outside of the cell, and so forth. So it's kind of like this really important border between the inside and outside of the cell that protects the inside of the cell from things that shouldn't be in it, keeps the things inside the cell that should, and prevents everything from just mixing and becoming completely chaotic, basically, because remember, the laws of thermodynamics tell us that things tend towards entropy. And so the membrane is really preventing our cells from undergoing entropy, keeping them organized and compartmentalized. Selective transport. So again, it keeps things that should be in, and keeps things that should be out, out. But that means that there has to be ways to allow the right things in and out. So this is transporters, channels, pumps. We'll talk about diffusion, but obviously it's preventing the wrong things from getting in. This is where we start to get to things that are a little bit less obvious. So the membrane is actually a scaffold. So things can be localized together by being in the membrane, or at least attached to the membrane. So think about this. The cell is this huge three-dimensional volume, aqueous-filled aspect, right? And so if you need proteins to come together, to work together, that, if it has to happen by diffusion, it works, but chances are much less likely for those two things to interact together if they're in this large three-dimensional space. If they're already hooked together in the membrane, yes, there's movement there, but they're going to be a lot more likely to come together. All right, so you can think about this in terms of just meeting up with someone. If you told someone that you are going to meet them at 12:15 on campus, well, campus is a big area. But if you told them that you're going to meet in Roar Science, then that narrows that down. Or if you and someone else were both out in the middle of the ocean, you're much less likely to come in contact with each other than if you're stranded together on the same island. Right? So membranes can bring these proteins together and allow for much more efficient signaling cascades along the way, from one thing to the next, to the next, to the next, because they're already in relative proximity to each other. And then we have signaling here. So inside-out signaling is when something inside the cell gets sent as a signal outside of the cell. So this is often in terms of exocytosis, releasing ligands. But then there's the outside-in signaling when a ligand binds to the receptor, causes a change inside. And then the membrane also is involved in those cell-cell interactions, adhesions, all those things as well. And then this is probably the one that's hardest to think about, but energy transduction. So the oxidative phosphorylation, right, that part of the mitochondria, that part of cellular respiration happens across the membrane. And there's a reason for this, because moving things across the membrane involves energy. So if you're moving things from low to high, that takes energy. Anything moving from high to low then gives off energy. And so the mitochondria are using the energy from NADH, FADH2, all these things that were made through cellular respiration. They're taking that energy and they're using that energy to pump protons against the gradient. And then when those protons come back with the gradient, that releases energy that's used to convert ADP to ATP. Right? So that membrane is really important for that energy transduction. But compartmentalization is as well. So the mitochondria have two membranes, and the reason for that is because you're pumping protons and you're creating a gradient between the outermost membrane and the inner membrane. That means that's a much smaller space. And in a much smaller space, you don't need as many ions to make the concentration high. If they had to pump ions into the whole cell, right, into the cytoplasm, you would have to pump a ton of ions just to create a high enough gradient for the protons to flow back into the mitochondria and create that ATP. So the fact that they are pumped into this relatively small compartmentalized space between two membranes means that you don't have to pump as many to have a really strong gradient and bring it down. Think about it for a second. If you were trying to pump water to raise the volume of water, it's much easier to pump water into a pond to raise its volume because the total area is much smaller than it would be to pump water into the ocean and expect that to be higher. So those membranes kind of compartmentalize, make a smaller area. You can get energy because you can create a faster gradient. All right, so moving on to things that we definitely know and hopefully are review, but this is a phospholipid. So phospholipids, basically you have the glycerol here with 3 carbons. 2 of those glycerol moieties are bound by fatty acid chains, right? So this is the 2 tails of the fatty acid, 2 fatty acid tails of the phospholipid, sorry. And then we have the other end, which is the phosphate group, hence the phospholipid, and some head groups that are bound to that. And these head groups can be quite different. The fatty acids actually can be quite different too, not only in terms of double bonds versus, so unsaturated versus saturated, but also their lengths and things like that. So here's kind of that phospholipid in a more three-dimensional image along the way. And then this is just kind of that cartoon simple representation. So because of this, we all know this from earlier courses, but because of this, the phospholipids will automatically form a bilayer. And the reason for that is those fatty acid tails want to get away from water, right? So these fatty acid tails, hydrophobic, want to get away from water. The phospholipid head groups are polar, and they're fine interacting with water, so the head groups end up on the outside and inside, both of which are aqueous, and the fatty acid tails of the phospholipid are in the hydrophobic interior of the phospholipid bilayer. So this happens completely spontaneously, and you can actually make liposomes as well. Liposomes are basically used for drug delivery, but these also can be thought of as vesicles for moving things inside the cell. And we'll learn all about vesicles in Unit 3. But why would you create a liposome for drug delivery? Well, for one thing, you can put a polar drug inside the liposome, and this phospholipid bilayer can fuse with the membrane bilayer and therefore release the polar drug inside the cell, just like a vesicle fusing with the membrane during endocytosis and exocytosis. This also can be very useful for nonpolar drugs. So if you put a nonpolar drug and it has to get through the blood system in order to get to the right cells, it's going to crash out in the blood system. The blood system is aqueous. But the nonpolar, the hydrophobic drug, can go inside this phospholipid bilayer, and it's happy there, and then the liposome can deliver it to the cell, and you'll still might have a little bit of a hard time because it's going to be stuck in the membrane of the cell itself, but oftentimes you can have this at high enough concentration that the drug will still end up doing its job. And you can add things to the membrane or to the outer edge of these liposomes that allow it to target to the right cells. So if you have an antibody that only binds to lung cells, you can deliver a drug to the lung cells, and so forth. You can kind of do this with all kinds of targeting markers. All right, so that's kind of how you can use that phospholipid bilayer to your advantage and create liposomes for drug delivery. And it's the same spontaneous formation that happens naturally for cells as well. All right, another quick review, fluid mosaic. All right, so the fluid mosaic model, quickly define what that means to you. So we have the fluid mosaic model, and this basically means, mosaic means there's a lot of different things in there, so we'll learn a lot more about this too. But we have proteins, we have carbohydrate residues on the outside of proteins or lipids, we obviously have the phospholipids, we have cholesterol, and we have the proteins that can go through the membrane in various ways. It can be partially through the membrane. This is a mosaic because the membrane is composed of a lot of different things. And it's fluid because these things are moving all around. So, my next question for you is, what movements are there? So which movement do you think is the fastest? We have 3 types of movements. So first, go ahead and see if you can identify the 3 types of movements. All right. So we have rotational movement. So these phospholipids here can just rotate around very rapidly. The membrane proteins can rotate. We also have lateral movement, so they can move side to side. They're exchanging positions very, very rapidly. And the membrane proteins, unless they're anchored somewhere, also move side to side very rapidly. What they don't do is flip from one leaflet, so this would be the inner leaflet of the cell. This would be the outer leaflet of the cell, extracellular, cytoplasmic. They're not going to go from here to here. The reason for that is, remember, this is a completely enclosed membrane, right? This is 3-dimensional. It's not necessarily a sphere because they take on all types of shapes, but they are fully enclosed. So that means that in order to get from this side to that side, this polar head group would have to go through the hydrophobic interior and then off to the other side. And that's not going to happen very often at all. And if it does happen, the cell needs it to happen, the cell has to use an enzyme or a protein called flippase to help it move because of that hydrophilic properties. All right, so which of these are going to move the fastest? We have the 3. We have rotational, we have lateral, then we have flip-flop. I already said flip-flop rarely occurs. How fast do you think these things are going? Well, one of these can rotate every 10 to the negative 9th second. So another way to put that is each phospholipid or each membrane protein, whatever, can rotate 10 to the 9th times per second. That's a ton. They can also laterally diffuse 10 to the 6th positions per second. Right? That they're moving laterally extremely fast. So I really have a hard time thinking about this. So I kind of did a little bit of mathematics. I know the size, the general size of each phospholipid. So if they're moving kind of this distance, they're moving about 10 to the 7th positions per second. Right? That means they're moving 5 to 10 microns per second. Now that actually doesn't sound like a ton, but when you're the size of a phospholipid, that's moving extremely rapidly. Right? So let's scale that up. So that means that each lipid is literally moving 36 miles per hour in your microscopic level membrane. So in perspective, if this were a car, all right, so if we made that phospholipid to the size of a car, something we really can kind of start to understand speed, that means the car would have to be moving 684 million miles per hour to move the equivalent speed of the phospholipid based on size. So this lateral movement is happening extremely, extremely rapidly. On the other hand, flip-flop, basically one phospholipid. Now remember, there are trillions and trillions and trillions of phospholipids in your membrane. One will move about per day, randomly, just accidentally flip-flop. So it's almost never happening unless the body has an enzyme to do it. And so here's a video that kind of shows some of that movement.
To demonstrate the fluidity of the lipid bilayer, a piece of the plasma membrane of this neuronal cell is pulled out with laser tweezers.
So they've actually grabbed this using laser tweezers, which we talked about on
Remarkably, moving this
Monday.
membrane tubule rapidly back and forth does not rupture the plasma membrane, which flows quickly to adapt to the mechanical distortion.
So basically, as this membrane moves around, even though they've pulled that out, the phospholipids are moving rapidly to compensate for this, and they can move so rapidly, they can spread out, they can change basically their interactions to the extent that that's not going to break. And this lateral, this lateral movement of those phospholipids, so the lateral movement of the membrane allows the membrane to do all kinds of three-dimensional forms. It can send out all these filopodia. This is why I say that a cell is not really a sphere. The membrane will be completely enclosed, but it is protruding, it's moving, everything's moving extremely rapidly. So, what we are going to do is, in a minute, we're going to do a project with bubbles to really learn the extent of this. But first I want you to think about, so why do the membranes need to be fluid? Right? We need partial fluidity. They can't be completely fluid, but they do still need to be fluid. What are some of the reasons for that? Think about it and see if you can answer so far. All right. One, we need mobility. So those membrane proteins, those things, they need to be able to move for a lot of reasons, which we'll talk about later. But for example, two monomers in the membrane, two membrane proteins that are part of a dimer, must come together. And in order to come together, they have to be within a fluid membrane. Then they might need to go pick up other things or signal other things. So those membrane components do need to be fluid. And we'll talk about how you can even regulate their fluidity. Not all things are moving as much as others. We also need to be able to make and fuse vesicles. All right, so during protein transport, things are going to be transported through these vesicles. So the vesicle has to bud off a membrane and then fuse with another membrane. And this is really, really important. Formation of organelles, all kinds of things. Cell movement, we just saw this video, but in order for the cell to move, the membrane has to move with it. Okay. But we can't be completely fluid because basically we need some structural organization. If we were completely fluid, or the cell membrane was completely fluid, that is, it would basically just be like water, and it would just kind of fall apart. So it needs to be held together, and it's held together because of those hydrophobic interactions, amongst other things. But it also needs to be fluid so that the cell can do all its things, its dynamic nature. So let's talk about how to really visualize this. And this is where we get to do something fun. We would do this together in class, but at this point you guys all know bubbles, so we can do this. So you can either buy a big bubble solution, or you can make some. This is the recipe I use: 2 liters of water, a 1/4 cup of dish soap per your 2 liters. Dawn seems to work really well, but almost any dish soap will work. A 1/4 cup of corn syrup. This adds some thickness. And then, so you're going to mix the corn syrup and water, semi-slowly mix in the detergent. If you don't seem to have kind of enough bubbly bubbles, you can add a little bit more detergent. It really depends on how thick your detergent is anyway. But you can make bubbles, or you can just go to Amazon or your local store and buy a, big, huge thing of bubbles for a couple bucks and go for it. All right, so now why are we talking about bubbles when we're talking about phospholipid bilayers? Well, bubbles are basically an inside-out phospholipid bilayer. So we've talked about the phospholipid bilayers. You have the polar head groups on the outside and the hydrophobic tails on the inside. That's because our cells are in an aqueous solution. We are made of water. So the inside of our cells, the cytoplasm, is aqueous. The extracellular regions of our cells are aqueous. And so these polar tails are happy interacting with water on the inside and outside of our cell. And that makes the hydrophobic tails basically try to get away from water and go to the membrane on the inside. Well, bubbles aren't in an aqueous solution, bubbles are in air. And air is basically hydrophobic. So the detergents in our bubble solution are micelles. All detergents have a polar head group and a fatty acid tail. Right? The reason for that is because they can then kind of grab those fats or those oils or things like that that are on us, and they help wash it away with the water. But those detergents, that polar head group and the fatty acid tail, well, in bubbles, the fatty acid tail is happy interacting with the hydrophobic air. So the air on the outside and the air on the inside of the bubble, those turn inward, or turn towards that. And then since we mix our detergent with water, there's water trapped between this. And so now the polar head groups are in the inside where the water is. So again, this bubble is basically a phospholipid bilayer. Instead of phospholipids, we have detergent micelles, but they are inverted. But the properties are going to be very, very similar, and that's the properties of movement and all kinds of things. And so we can really learn about the movement of our phospholipids, and we can kind of visualize a lot that's going on really by using bubbles. All right, so go ahead and go to the next video and I'll explain what we're doing. And this it can be a fun project. Like I said, we would normally do this in class, but with bubbles, I think you can do it on your own. And you can even submit a little video of what you've been doing for a little bit of extra credit. So hopefully we can have some fun with this. So I'm going to go ahead and stop this video now and then move to the next one that talks a little bit more about the bubble project.